Hello, and thank you for attending my talk on the Ulukhaktok Fish Telemetry Program. I'd like to acknowledge my mentors and co-leads on this project, Nigel Hussey and Lisa Lazzetto, and collaborators from across the country, including the Ulukhaktok Hunters and Trappers Committee and the Char Working Group. Today I'm going to talk to you about the project as a whole, including multiple student and postdoc projects that involved in this program. The Ulukhaktok Fish Telemetry Program was based out of a community on the west coast of Victoria Island, Northwest Territories. The region has seen unusual events and food web changes recently that are thought to associate with climate change. Through several consultations and meetings with the community and working groups, we developed research questions that focus on species, study sites and topics in line with community and regional interests and concerns. Our main objective was to investigate species capacity for flexibility to climate change effects through individual specialization in mobility and feeding ecology on Arctic marine coasts. Our study species were Greenland Cod and Arctic char as two species with contrasting life histories and functional ecologies to give us a broad understanding of species potential response. Our methods include an extensive acoustic telemetry array to look at movement ecology, whereby we tagged fish with internal transmitters and deployed benthic moorings to detect the fish as they swam past. Moorings were deployed in a fine-scale 5-kilometre grid and sparsely along 300 kilometres of coastline near the community. We used a portable receiver to detect char when they returned to overwintering lakes, and to measure potential drivers we used oceanographic loggers on moorings and conducted habitat and food web surveys. We examined the prevalence of individual specialization in trophic ecology from stable isotopes of carbon and nitrogen across multiple tissues to indicate how individuals shifted their diets over time. And we looked at variation in body shape using geometric analysis of photographs to identify polymorphic individuals. Artichara had divergent marine migration and residency strategies depending on the overwintering lake they returned to. For example, Arctic char associated with Hikiak Lake and the Cook River on the far right in Prince Albert Sound spent a lot of time in the semi-enclosed safety channel, whereas char associated with the Gujra River in Minto Inlet to the north did not and spent more time near Ulukhaktok, which is important for understanding fishing pressures on these different populations. In relation to trophic and habitat shifts with body size, indicated by change in delta 15N and delta 13C stabilized between fast and slow turnover tissues, anadromous Arctic char had high inter-individual variation in size-based trophic shifts and habitat shifts during spring-summer. Incidence of variation within and among individuals increased with body size. From geometric analysis of photographs, we found 3 polymorphs of Arctic char in symmetry on the marine coast during our summer collections, driven primarily by head morphology, which likely associated with the intra-individual variation in feeding ecologies and habitat use on the marine coast. These combined findings indicate that Arctic char are highly plastic to changes in prey availability, which may depend on access to optimal habitats for summer foraging. Their high mobility and individual specialization with ontogeny provide them with capacity to do so. We also found measures of body condition correlated with greater trophic and habitat flexibility, highlighting potential payoff that flexible foraging provides. For the more sedentary perennial marine resident Greenland cod, we found low population level variation in activity, space use and distance travelled throughout the year, with minimal movements over a small distance for most individuals. Though some relative extreme examples of distance and activity space use were found throughout the year among a few individuals, indicating possible individual specialization. When movements of cod did vary, it was driven primarily by changes in salinity, with larger space use and distance travelled at lower, more brackish salinities and lower ice concentration. The random component, which is the fish ID, had a much greater effect with 50% depending on the metric compared to 8% for all environmental and demographic variables combined. This high unexplained variation within and among individuals also indicated capacity for flexibility for this population. For sterilized data in Greenland Cod, we found that intraspecific variation in trophic and habitat shifts did not increase from juveniles to adults, and the shift was consistent among sizes in the sampled population. Correcting for body length, variation in body shape indicated presence of two morphotypes within the small coastal area. These findings show that high within and among individual variation in movement and feeding ecology suggests capacity for flexibility at small geographic scales, with small distance movements supporting a generalist diet across sizes. So to conclude on the multiple components of the Ulukhaktok Fish Tagging Program, noting their physiological and functional differences, both species exhibit capacity for flexibility, with examples of individual specialization in marine habitat use, ontogenetic trophic and habitat shifts, and multiple morphotypes in sympatry. Both species may be dependent on heterogeneous coastal marine habitat hotspots. Arctic char during summer and with favoured location depending on overwintering lake, and cod throughout the entire year. In comparison with other literature, these species could be experiencing initial benefits of the longer open water season for feeding and rapid growth, but once conditions reach physiological limits, Greenland cod may move offshore and Arctic char may become increasingly limited to fresh water, which would have implications for coastal fisheries management and maintaining sustainable subsistence harvest. To address these broader questions for this region will require collaboration among multiple groups and communities in the region. Thus, the project is now transitioning into cross-community research and capacity building to address questions in relation to marine protected reserves. So thank you for listening. I'd like to say thank you again to our funding partners and collaborators, and I invite you for questions.